Okay, so thank you for that introduction, Dr. Fuhrer. Today I'm going to be speaking about my thesis work, which is on profiling ductal carcinoma in situ and invasive ductal carcinoma using desorption electrospray ionization. Oops. Okay, so breast cancer is the most common cancer among women, with 1 in every 8 women to be diagnosed, which amounts to about 29,000 new cases in Canadian women each year. 1 in 36 of these women will unfortunately die due to this disease. And of the 29,000 diagnosed, about, oh, we lost my graph, but that's okay. About 20 to 25% of those are DCIS diagnoses, and that translates to up to 7,300 new DCIS cases per year in Canada. So despite advances in early detection and novel treatments, there's still work to be done to prevent breast cancer-related deaths. So on the left, oops, just gonna add my laser pointer here. On the left, to help orient you, is a cross-section of the breast, reminding you of the lobular and ductal features of the mammary gland, as well as the proximity and interactions that happen within the surrounding stroma. So ductal carcinoma in situ, or DCIS, is a form of non-invasive breast cancer that presents itself as an abnormal epithelial cell population that is contained to the ductal system of the breast. An invasive ductal carcinoma, on the other hand, which is known as IDC, occurs when ductal carcinoma has grown through the ducts and has begun to invade surrounding tissues. It's important to acknowledge that pathologists classify DCIS into low, intermediate, and high grades based on some of the pathological features I have listed here. So low-grade DCIS can often resemble this cribiform pattern in which abnormal cells have begun to grow into the lumen. And high-grade DCIS can be characterized by this solid filling of the lumen, which can even lead further into necrosis. IDC, on the other hand, the cells have left and move out of the ducts, and it's a much more chaotic environment. So while DCIS can present with physical symptoms such as a palpable lump or nipple discharge, it's most commonly detected through routine mammography screening. And translating its detection to ensure complete surgical removal can be one of its clinical challenges. Although women with DCIS are generally given a good prognosis, there's great uncertainty surrounding the risk of DCIS recurrence, and we're currently unable to predict the risk of tumor invasiveness. So due to these knowledge gaps, many women often opt for aggressive therapies, such as a full mastectomy or segmental resection with radiation. And unfortunately, this aggressive overtreatment of many patients can lead to physical and mental distresses, surgical comorbidities, and just an overall lower quality of life. So more work is needed to overcome these challenges, and the path that we have chosen to examine is the metabolome. So there are a large number of breast studies that have looked at DNA mutational as well as transcriptomic data of various types of breast tumors to uncover patterns that will provide diagnostic and prognostic features to help patients. However, there are still many challenges in this field, and my focus is going to be on the metabolome. And the metabolome provides a profile of cellular activity and small molecule signaling, such as sugars, nucleotides, amino acids, and lipids, and how they affect the phenotype and function of a cell. So metabolomics is an emerging field of study that lets us assess cellular changes in the metabolome, but also integrates and reflects upstream influences of the genome, the transcriptome, and the proteome. So next, I wanted to talk a little bit about the importance of lipid metabolism in a normal cell and how tumor cells can alter that lipid metabolism. So this is a busy figure, but it reminds us of the many different pathways that are involved in normal lipid metabolism processes. So for example, we have fatty acid oxidation, which can lead to fatty acid synthesis, responsible for producing phospholipids that are important for the formation of cell membranes. We have pathways involved in the production of arachidonic acid, followed by pro or anti-inflammatory eicosanoids, which I'll go into more detail about on the next slide. But the takeaway here is that all of these various lipids that are important during normal physiological processes are typically under tight regulatory control. However, rapidly proliferating tumor cells can have additional lipid requirements that can exploit many of these pathways for their needs. So, for example, increasing extracellular lipid uptake and phospholipid production in order to account for those rapidly proliferating cells. Tumor cells can create a pro-inflammatory environment through the increased metabolism of arachidonic acid. And so what I'm getting at here is these fatty acids, such as arachidonic acid, they're recognizable. And when there are increases or decreases in these fatty acid levels in tumor cells, we can detect that through the use of metabolomics to distinguish how tumor and normal cells are behaving. So as an example, I want to focus on arachidonic acid metabolism, as this is a well-characterized pathway that is regulated by cyclooxygenase 1 and 2, as well as lipoxygenase, and it generates a number of different downstream lipid metabolites, such as prostaglandins and leukotrienes, that can be both pro and anti-tumorigenic. So one example that I want to highlight is PGJ2 at the bottom here. And it's a prostaglandin that's associated with anti-tumorigenic activity. And interestingly, it also serves as an endogenous agonist of a nuclear transcription factor known as PPR gamma. In contrast, PGF2 is a prostaglandin associated with pro-tumorigenic activity, which activates genes that shut down PPR gamma. And so knowing the profiles of these fatty acid metabolites, as well as how they're being used in tumor cell metabolism, helps to put into context what might be happening in the cell, and it's useful to link these pathways back to see what's active in DCIS and IDC tumors. So one of the novel ways we are examining the metabolomic profiles is with a method known as desorption electrospray ionization, which I'll refer to as DESI. And DESI is a rapid, non-destructive mass spectrometry imaging technique that allows for the detection of small molecule metabolites and lipids of a tissue sample at ambient temperature. So DESI is typically conducted on fresh frozen tissue. It requires very minimal sample preparation, and it characterizes its samples based upon its mass to charge ratios. So this is the DESI sprayer, and I've kind of created a cartoon version here. So in DESI, a high velocity ionized solvent is sprayed directly onto the sample surface of a slide, and that desorbs the analytes directly from the slide into its gaseous droplets that are then delivered to the mass spec inlet where those mass to charge ratios are analyzed. So these are the types of 2D heat maps that DESI produces. And for each 100 by 100 micron area we see on this DESI slide, we generate an individual spectra such as this. So as well, because DESI retains its spatial resolution of the data, it allows the locations of the detected ions to be analyzed in comparison to the gold standard histopathology defined tumor and non-tumor regions. So this means that we know where the data is present on any given part of the slide. So there's not that much that is known about the signatures of DCIS. If we look to the literature, there was a study done by a group in Brazil that used DESI to assess the metabolome of various breast cancer molecular subtypes. And within their cohort, they happened to have about 15 DCIS samples, and they compared those to invasive breast cancer as a whole. They found that there was an upregulation of glycerophospholipids that was detected in DCIS compared to invasive breast cancer. And they thought that this may be related to apoptosis mechanisms. And in terms of invasive breast cancer, they saw increased expression of docosanoid molecules. And they thought that that might be related to inflammatory mechanisms. As a reminder, these are the types of fatty acids that are generated during the arachidonic acid metabolism pathway and things that we can see with the DESI. Another paper examined markers associated with DCIS recurrence, and they saw that increased COX-2 expression was associated with increased DCIS recurrence. And so they also showed that increased expression of PPR gamma, that nuclear transcription factor that I'll speak more about in a few slides, was associated with lower grade DCIS and less recurrence. So this suggests that PPR gamma expression might be protective against DCIS recurrence, but together, these limited studies that are kind of the only known research in this area suggest that the fatty acids generated in the arachidonic acid pathway may be useful to help obtain prognostic information about DCIS and IDC samples, and that PPR gamma signaling may be protective. So PPR gamma, what does it stand for? It's the peroxisome proliferator activated receptor gamma. It's a ligand-activated nuclear transcription factor, and it's expressed in many mammary gland epithelial and associated stromal cells. PPR gamma is activated through endogenous lipids, including prostaglandins and fatty acids, as well as synthetic thiazolidine dione drugs, such as rosiglitazone, which I'll refer to as Rosi. And so these ligands induce a conformational change, which allows PPR gamma to form a heterodimer with the retinoid X receptor, resulting in increased transcription of its target genes involved in normal lipid and sugar metabolism. Most importantly is that PPR gamma plays a role in suppressing the growth and spread of breast tumors, both in vitro and in vivo. So as I mentioned, work like this showing the in vitro and vivo work has been done by others as well as members in our lab, and it's shown that PPR gamma can impact many signaling pathways in addition to the lipid metabolism pathways I referred to earlier. So this is a non-exhaustive representation of some of the PPR gamma pathways that protects against breast tumor progression. But in the context of breast tumors, we know that PPR gamma activation exerts an anti-breast tumor progression/metastasis effect. So this is a busy slide, but there's a couple of things I want to note. First, that activating PPR gamma signaling can increase tumor suppressor expression such as BRCA1 and PTEN, which in turn can decrease proliferation and increase apoptosis. I previously mentioned that another group has associated COX-2 with increased DCIS recurrence and PPR gamma with decreased recurrence. And this is interesting as PPR gamma activation actually decreases cyclooxygenase activity. So perhaps PPR gamma could be playing a protective role in that transition from DCIS to IDC, or even from low to high grade DCIS. And lastly, PPR gamma can downregulate cyclin D1 expression, which in turn can decrease cell proliferation and cell cycle progression. So what we don't know right now and what we're trying to figure out is what are the metabolite profiles that distinguish DCIS from IDC, and does PPR gamma prevent DCIS progression? So I've hypothesized that DCIS and IDC tumors have distinct DESI profiles reflective of signaling pathways that protect against tumor progression. And to assess this hypothesis, I have three aims. The first being that I want to define the metabolomic profiles associated with human DCIS in IDC ex vivo samples using DESI. The second being that I want to determine whether a PPR gamma P10 signaling axis is protective in human DCIS in IDC-derived cell lines in vitro. And the third being that I want to develop a preclinical in vivo xenograft model for DCIS and IDC to assess clinically relevant targets from aim 1 and 2. So this is an overview of my first aim, which is centered around the human feasibility studies. And I'm going to go into more detail about my cohort in a minute, but with the help of Dr. Varma, I was able to collect 20 FFPE, so formalin fixed and paraffin embedded DCIS and IDC samples from the Kingston Health Sciences Center. And because these samples are formalin fixed and paraffin embedded, I needed to deparaffinize them before I could DESI them. After they were DESI'd, that same slide that was DESI'd was then taken for H&E and annotated by a pathologist for its tumor and non-tumor regions. And then with all of that data, I was able to perform my data and pathway analysis and then try to relate some of these clinical correlations. So a bit about my cohort. This is a prospective human breast cancer cohort, and all these samples are from the Kingston Health Sciences Center. I have 20 samples in total, and of that, I have 7 DCIS samples, 3 IDC samples, and 10 mixed, and that means for the mixed samples that both DCIS and IDC were present. As I said, they're FFPE samples, and working with Dr. Koester and Dr. Varma, we have identified 11 pathological zones that we think would be important to assess using DESI. And what I mean by pathological zones, we looked at DCIS, IDC, adipose, benign glands, etc. So next, I'm going to briefly go over my data analysis pipeline because I think in order to understand the results I'm going to present, we need to understand the mass amounts of data we're working with. So the first thing I do is I visualize the pathological zones that were annotated by Dr. Koester. And then I look to the DESI images and try to find some images that help me orient myself between the DESI and the H&E. I then overlay the H&E image with the DESI image, and I take my time with this so it's as precise as possible. And then I can plot regions of interest, which I'll refer to as ROIs, that correspond to each of those pathological zones. If you remember, I said a bit earlier that when I DESI in 100 by 100 micron pixels, and each of these pixels contains its own spectra. So when I plot a region of interest, I'm going to merge 9 of those pixels together to form one region of interest. So I'm merging 9 spectras and I'm averaging all 9 of those spectras. The data that I pull out from those regions of interest is then normalized to the total ion current. And then because I'm doing this for every single slide, I have to align all my slides together. And then I'm able to perform my statistical analyses. So one of the first things that I wanted to do was I created a PCA LDA model using the 11 pathology defined zones. And what we're seeing here is first, we are looking at adipose in red. And then kind of as we move, we see a little bit of a metabolomic change to the benign connective tissue and stroma in orange. It might be difficult to see, but in yellow, we have the benign glands. And then DCIS here in this light blue, and IDC, tumor inflammation, and tumor stroma in this darker blue. Kind of scattered throughout, we have ROIs present in blood vessels, and cautery, as well as benign inflammation. And so when we look at the model performance of this, in total, I had about 23,000. Uh-oh.
You probably just your connection.
Ah, great. Uno momento. Oh, thank you. Great. So when we look at the model performance here, there was about 23,000 ROIs or spectras in total, and about 22,500 of those were classified correctly. 700 of them were not. So that left us with a model performance of about 81% for these 11 pathological zones. However, there were 6 of these zones that on a tumor slide, they just don't take up the same amount of square footage. So it's impossible to get the same number of ROIs. Like you can't compare an IDC tumor that we have compared to a couple of blood vessels we have speckled around. So it's very difficult to get the same number of data. So this data set was a little unbalanced with all of these 11 classes. So I decided to create a same model, but only with 5 classes this time. And I excluded the other smaller 6 classes. And so what we saw is, I'll just need my laser pointer back again. So what we saw adipose in blue, and as we move down, we see benign connective stroma in orange, benign glands in green, and then DCIS kind of smattered on top in red. And then the farthest away, which means it had the most metabolomic difference from the rest, is the IDC regions of interest. And so with this model, we had about 22,600 ROIs, and 22,300 of them were classified correctly, and 350 of them were not. Even have 350,000. And so that left us with an accuracy of about 91% for these 5 pathological zones. And so I wanted to take kind of a step further and break it down a little more and look at the confusion matrix as this breaks down how each ROI was classified. And I want to focus specifically on the DCIS ROIs. So in total, I had about 480 DCIS ROIs. And of that, 401 were classified correctly, which is great. But I had 25 that were DCIS that were predicted to be IDC, and I had 27 DCIS that were predicted to be benign glands and ducts. So given that these ROIs are selected based on the gold standard histopathology of the sample, my data may suggest that DESI may be detecting metabolomic changes in DCIS progression that's not visible with histology. And so I'm going to be consulting the pathologists on these to further discuss the potential implications. But once this is confirmed, we may be able to enhance the information available to pathologists during their classifications. And this is just one example of how DESI profiling may improve pathology decision-making. So while this is all good and dandy, I was really excited about looking into the particular ions that were up or down regulated in DCIS and the biological role that they were playing. Sorry, water break. So this is a volcano plot that separates binary data on the basis of statistical significance as well as increasing fold change. So on the right in red, we have ions upregulated in DCIS. And on the left in this bluish purple, we have ions that are upregulated in IDC. And so I have assessed the entire mass range, but for the sake of time, I'm going to show you the results I'm most excited about, which take place in the mass range of 3 to 600. And this is typically where the fatty acids reside. So first, I wanted to take a look at the ions that are significantly elevated in IDC. So these are just a handful of ions that are upregulated. And using the volcano plot, I separated out the data that was statistically significant, as well as had a fold change of at least 1.5. And so this brought me to the series of ions that are highlighted in purple here, which contain arachidonic acid, as well as some of its up and downstream counterparts, including gamma-linolenic acid and docosahexaenoic tetranoic acid. And so these bar graphs show how each of these ions highlighted in purple are very significantly elevated in IDC compared directly to DCIS. And as I was reviewing these ions, I realized that the majority of them were omega-6 fatty acids, and that there were other omega-6 fatty acids that had not come up as having a fold change of at least 1.5. So I went searching for them to see if they were at least statistically significant. And they were, especially linolenic acid. And so I was surprised, yet very excited, that I was looking at quite a few omega-6 fatty acids here, which can be inflammatory and brings us back to that increase in fatty acid synthesis I was speaking about before and the involvement of arachidonic acid. So it's important that we're seeing omega-6 fatty acids for a couple of reasons. One being that omega-6 fatty acids are pro-inflammatory and can promote invasiveness and progression, which is seen in IDC. All of these fatty acids in the omega-6 family are significantly upregulated in IDC, so it's not just a couple. And so starting with linolenic acid, and we move our way down to arachidonic acid, this here is where we're really going to be, where we might be seeing a lot of those pro-inflammatory eicosanoids coming out, contributing to a pro-inflammatory environment in IDC. But the second reason that these ions being elevated is exciting is because of the ions I saw elevated in DCIS. So here I wanted to discuss two ions that I found upregulated in DCIS compared to IDC, independently of the ions I just discussed. So the first was 15-deoxyprostaglandin J2, which is one of the anti-inflammatory prostaglandins I mentioned earlier. And this ion was significantly upregulated in DCIS compared to IDC with a fold change of 1.6. So this prompted me to see if I could find any of the precursors to PGJ2, in which I found prostaglandin D2 to be significantly upregulated in DCIS as well. So I was trying to put these puzzle pieces together with arachidonic acid metabolism and its prostaglandins in DCIS. And so what we're seeing is an increase in PGD2 and PGJ2 in DCIS, both of which contribute to an anti-inflammatory environment. So the extra exciting bit here is, if you remember from the beginning, PGJ2 is actually a direct PPR gamma ligand. So to summarize my pathway analysis from the DESI signatures, my analysis suggests that these pro-inflammatory signals are more prominent in IDC, and that these anti-inflammatory signals are more prominent among DCIS. As PGJ2 is a ligand of PPR gamma, it's possible that we're seeing an anti-inflammatory effect in DCIS due to PPR gamma activation, and potentially reducing or slowing down the progression from DCIS to IDC. More work is definitely needed to confirm this, which leads me to my next slide, in that for my ongoing work of this aim, I want to assess the PPR gamma and COX-2 expression on these human sample slides with IHC staining. I also want to validate these ions' identity using fragmentation. I want to increase my sample size to hopefully increase my model testing and cross-validation, and I want to correlate these results with clinical features. Something that I haven't spoken about here, but is an exciting part of this work is that, as I said, DESI is typically conducted on fresh frozen, but it is very difficult to obtain fresh frozen breast samples, never mind DCIS. Anything that is suspected to have DCIS is put into FFPE for pathology diagnosis. So we were only able to obtain FFPE tissues right now, but we've done some work and we've been able to obtain a small cohort of fresh frozen. And so what I'm working on right now is trying to validate the signals that we're seeing in FFPE tissues with DESI by comparing it directly to its fresh frozen counterparts from the same patients. So this brings me to my second aim, which is focused on my in vitro work. And so here we're looking at the two cell lines that I'm using. So they're both human breast cancer epithelial cells. The first one are the UACC812 cells, which are representative of DCIS. The second are BT549 cells, which are representative of IDC. So the 812 cells are estrogen and progesterone receptor negative, but have an overexpression of HER2. They are wild type for PTEN expression, and they have low PPR gamma parental expression. The 549s are triple negative, and they have a mutated PTEN expression and low PPR gamma parental expression as well. And so what I'm doing in my in vitro work is I have both of these cell lines and I have transduced them with the WPI luciferase as well as an inducible PPR gamma wild type plasmid. And from there, we looked at baseline protein expression, and then I was able to conduct some of these phenotypic assays to assess PPR gamma's activation in invasion, proliferation, migration, apoptosis, and tumor spheres. And then from there, I was able to complete my data and pathway analysis. So what I want to discuss here today is the proliferation results of my 812s and 549s with and without the expression and activation of PPR gamma. And so first, I'm going to focus on the DCIS cells, which are the 812 cells. And so this graph is representative of the growth rate over time, looking at 24, 48, and 72 hours. And as I've noted here, the bars on the left-hand side represent the parental cells with increasing concentrations of ROSI from 0.1 to 10 micromolar, which represents the therapeutic range. And then looking at the parental cells, while there was an increase in proliferation, there's no net effect with these treatments. However, remember that these cells have very low PPR gamma expression. However, looking at the induced PPR gamma cells on the right sides of the graph, with activation, with ROSI treatment and PPR gamma activation, we see a significant decrease. Oh no, not again. I need to stop doing so many hand gestures. Okay. What was I saying? Oh yeah, induced cells. Okay, so looking at the PPR gamma induced cells with activation of ROSI, we see significant decreases in the proliferation, maximally so at the 48 hours. My laser pointer again. And it seems we might be seeing a bit of a rebound effect by 72 hours. And so similarly, when I performed this assay on my IDC 549 cells, the parental cells had very minimal response to ROSI at the 24-hour time point. However, there did appear to be a trend where 10 micromolar of ROSI reduced the proliferation and was significantly reduced by 72 hours. More interestingly, when we induced PPR gamma, we see more of a significant decrease in proliferation at 42 and 72 hours. So I'm currently working to further identify PPR gamma dependent signals that may explain these results, trying to center it around my previous findings from AIM-1, as well as the literature suggesting the PPR gamma P10 signaling axis. So here's a tumor sphere assay with the DCIS 812 cells, and my plan is to repeat this assay, but I did want to note that other than seeing, so this is the graph that goes with it, these are the parental cells and these are the induced cells. And so other than seeing this unexpected change with the vehicle in the parental cells, there was really no effect on tumor sphere diameter with ROSI treatment. But in the 812 cells that had induced PPR gamma expression, treatment with the 3 concentrations of ROSI significantly decreased tumor sphere diameter, which we can see by these representative images here. And so I'm working on completing this with the 549 cells and trying to understand the mechanism behind it. However, it's encouraging that the expression and activation of PPR gamma signaling reduces the ability of my DCIS cells to form a 3D mass, suggesting that PPR gamma may be protective. So my ongoing studies for this is to complete the rest of those phenotypic assays I discussed, as well as look at the expression changes in these cells to try to determine the mechanism that's occurring. And lastly, my third aim is on my in vivo studies. And so currently there's a handful of preclinical DCIS models. Most of them involve patient-derived xenografts, although there is one model that uses human breast cancer cell lines. However, it's very invasive. So what I sought to do was establish a novel preclinical DCIS model that would allow me to validate my findings in vivo. And so what I'm doing in my third aim here is I'm taking my DCIS cells, I'm injecting them intraductally into mice, and then because I've labeled them with the luciferase reporter, I'll be able to track them using luminescence. And then after that, after we excise the tissues, it follows a very similar pattern to my first aim, in which we take the slides, we deparaffinize them, DESI them, annotated analysis. So this, as I said, this model is based on previously established methods. We just modified it to make it less invasive, and we intend to characterize the progression in the model as well as the cell concentration. So what we do is we inject through the nipple of the fourth mammary gland to try and gain relatively restricted access to the ducts. And we used immunocompromised mice so that we can inject human DCIS and IDC cells. I chose a range of concentrations between 10, 20, and 50,000 cells, and I monitored these mice for up to 12 weeks. So we showed early on in our pilot studies that this technique is viable using trypan blue. So we injected trypan blue and immediately excised the mammary gland to see the solution extending through the branches of the ducts. And so what I'm doing to characterize this model is I injected 2 mice per cell line per time point. And then I monitored these mice each week using luminescent imaging. Every 2 weeks, I collected tissues, and we performed data collection, and we necropsied the fourth mammary gland where we inject the cells into. And then that fourth mammary gland is going to be used for DESI and pathological assessment. Our main goal here was to sample various cell lines at various concentrations at different time points to understand the profiles throughout DCIS progression and invasion. And as I mentioned earlier, by understanding the ions and mechanistic pathways of the human samples, we can use this xenograph model to further explore those pathways. And so this is an example of my DCIS cell lines at the three different concentrations. And so I wanted to show the growth with the luminescence. And so with the 812s, we're able to get tumor growth out to about 8 weeks before the mice reach humane endpoint with 10,000 cells. As we increase the cell concentration, we're able to see a quicker onset to reach study endpoint. And we're working to characterize these tumors histologically with my pathology collaborators, as well as to confirm the optimal time and concentration to generate these tumors that are going to mimic the pathology of human DCIS. And then we can proceed with a larger in vivo study. So my ongoing studies for my third aim is to repeat this with PPR gamma wild type induction and ROSI treatments using both of the cell lines. To analyze these tumors using DESI, and then try to correlate this in vivo work with what we found in aim 1. And so what we can conclude from my research so far is that we've optimized a method to use DESI on FFPE human tumor samples. We've established a PCA-LDA model that predicts human FFPE, DCIS, and IDC tumors with 91% accuracy. And we've seen that DESI profiles suggest pro and anti-inflammatory signaling may help enhance pathology characterization of DCIS. Expression and activation of PPR gamma signaling may protect against DCIS progression by decreasing proliferation and preventing tumor sphere growth. And lastly, we generated a less invasive intraductal xenograph model that may help with preclinical studies. So my long-term goals here are to integrate my 3 aims so that my analysis of my human DCIS and IDC tumors in conjunction with my in vitro and in vivo data will improve the prognostic information available to pathologists, which may be useful to expand treatment options for patients and ultimately improve their outcomes. So thank you. I just wanted to acknowledge everyone because there's been a lot of people who have made this work possible. Everyone in the Nickel Lab, of course, Chris himself, and my advisory committee, and then all of our collaborators who have been very key in making this work possible. So thank you, and I can try to answer questions. Thank you.
All right. I think Natasha would love to answer some questions.
I'd love to. Oh.
Oh, is there a question on Zoom? I don't know.
Hello.
We hear you. Go ahead.
Yeah, it's Sandip Singh Gupta. Thank you very much, Natasha. I enjoyed that presentation. So as a breast pathologist, we know that not all DCIS is the same. There are many architectural types, there's many nuclear grades, and not all of them have the same prognosis or likelihood, shall we say, of progression to invasive carcinoma. So, for example, the high-grade comedo types are the most likely to recur either as DCIS or as invasive carcinoma. So I might have missed it, but were you able to characterize the DCIS further to be able to determine which ones you're dealing with, so to be able to better sort of characterize which are the bad apples in the DCIS field?
So that is definitely something that I want to do. So out of the, I believe, 8 DCIS samples I do have the information of whether they are high or low grade, and I do want to try to correlate these signals with those features. I haven't yet, and part of the reason I haven't yet is because almost all of the samples I have are high or intermediate grade. It's been, oh man, not again. It's been really difficult to obtain any samples that are of a lower grade, so it's hard to make that comparison. But I haven't done it yet, but you bring up a very good point, and it is something that we do want to look at in the future.
I'm gonna step in here.
Sure.
So congratulations on getting that intraductal engraftment.
Thank you.
I imagine that was kind of tricky to.
Yes, it took a very long time.
So have you started looking at sections of those, and does it look like you're getting most of the cells confined to the ductal structure, and are you seeing invasive characteristics when you do the sections? And
yeah, so it's actually been very difficult to characterize it histologically, and it's something we're still working on because when we look at the mammary glands that don't have full-blown tumors yet, we're kind of having to shred the whole tumor block, the whole mammary gland block, to trying to be able to find those cells until we have a tumor. Kind of, like DCIS in reality, it's hard to find unless it's through mammography. So it has been very difficult trying to find those cells, but that is what we're working on right now, because histologically we kind of have a normal looking mammary gland until all of a sudden there's a tumor. So we know the tumor cells are there. We're just trying to locate them and to prove that they are in the ducts.
okay, thanks.
Yeah, it was a great presentation.
Thank you.
I have a couple of questions.
Mhm.
The first one is that I assume that the breast is a hormone responsive organ. So do you know what the hormone status was of these samples?
Yes.
So during the menstrual cycle or with therapy? And then, but maybe I'll stop there. I have one other question.
Okay. Yes, we do. I don't have that information right now. I think it'd be possible to see where these women were in their menstrual cycles. We have their ages, so we might be able to predict to a certain degree where they were. But I do have their receptor status of these tumors. And I haven't looked at it too far yet, because our kind of big goal at the beginning was to look at DCIS and IDC as a whole without taking those molecular subtypes into consideration. But as I move further with this analysis, that is what I'm going to be doing, is trying to break it up into our different molecular subtypes and make those comparisons for sure. So I do have that information. I just haven't gone there yet.
And maybe the metabolome will differ with the different hormones. So the other question I had was about, you showed evidence that you identified this group of inflammatory fatty acids. So the breast is made up of a lot of vascular tissue and adipose.
Mhm.
Did you see any markers of sort of showing inflammatory changes because of these inflammatory fatty acids? So increased vascularity, etc.
Yeah, so you're hitting all of the questions that I am hoping to do in the future. So one of the things that I didn't show here and I'm still working on is I wanted to see if there was a difference in these ions in that surrounding adipose, because the adipose is very important for like breast cancer and cancers that like to grow in adipose-rich environments, as well as the interactions that may be happening between the tumor cells and adipose. So I did wanted to check to see if there was an increase in those inflammatory ions in the adipose. However, all I have to work with is like the non-tumor regions. It's very difficult to characterize what it would be like in a normal human breast. So that is one of my next steps, is trying to compare it more to the surrounding non-tumor regions, not just DCIS to IDC. But yeah, just haven't gotten there yet. But you bring up really great points.
So in your two cell line models where you're topically overexpressing PPAR gamma,
Mhm.
and then using Rosie, have you been able to look at differential gene expression in those? And do you know to what extent you're getting an induction of genes that you should be getting?
Yeah, so we have looked at the induction over 6, 24, and 48 hours. And for both of the cell lines, with the induction of PPR gamma as well as with the activation. And we do see stable expression of PPR gamma for the DCIS cell lines. We see more stable expression through 6, 24 to 48 hours. For the 549 cell lines, we see the expression start to drop a bit after 48 hours. So it seems like the tumor cell is working hard on its homeostatic mechanisms to like bring it back down.
Yeah, I'm just
Sorry, what was the
wondering,
second part of the question?
target genes of PPAR gamma.
Oh.
See that it's actually.
Yeah, so
Interesting.
I'm just starting to work on those protein expressions, but we're looking at things like P10. And so very minimally, we've only done it once, so I don't want to say anything definitively, but we have seen like an increase in P10 expression with the activation of PPR gamma, as well as a decrease in cyclin D1 in the DCIS cells.
Oh, is that looking at protein or?
Yeah, that's looking at protein. Yeah. I wish I had those results to show you, but they're not quite ready for an audience yet.
Other questions? Might not get to hear her talk for another couple of years.
Oh, there's another one. I know. I can't see
You
the.
can't see the question.
Yeah.
That's Paula, go ahead.
Oops.
I'm here. Yeah, thank you very much, Natasha. That was great. I have a question related to what previous you have just previously been asked, but so for the AIM-1, the DCIS, you didn't really separate, or even for IDC, the different grades, or if there was chlamydia necrosis, like all the different things that pathologists look at. I can imagine that you didn't you didn't look either to the receptor status, specifically for DCIS. Usually, I don't know how old the samples were, but usually that was not done, is not currently done clinically, or until recently at least. For the AIM-2, you're looking at the worst kind of DCIS, which would be the equivalent, if you want to, of the triple negative of the invasive, right? Did I get that right?
It's our IDC cells that are triple negative. My DCIS cells are negative for ER and PR, but they overexpress HER2. So still a pretty bad version of DCIS, yes.
Okay, so it would be an overexpression of HER2. Okay.
Yes.
Why, can I ask you, why did you choose that kind of DCIS cell line?
Yeah, so quite frankly, they're just one of the only ones that are available for use right now. It's very difficult. There aren't really any cell lines that are available that are low-grade DCIS. In order to get a cell line like that, we would have to try to like make it from xenograft models or whatnot. But quite simply, it's just the only one that was available to us, and it happened to be a an aggressive form, and I think that's why it is a viable cell line, is because it is an aggressive form of DCIS.
Okay, so just for me to know, so that there is not really available, let's say, a DCIS cell line that is ER and PR negative. HER2 is not routinely done in clinical practice for DCIS, usually in many labs, in most labs at least. So the HER2 positive or overexpression now changes, as you said, into a more aggressive type of DCIS. But it's really hard to find at least just ER negative, PR negative without knowing the HER2. Okay. So
Yeah.
you're looking at two different molecular subtypes theoretically in the AIM2 between the DCIS and the IDC. Like one is the triple negative and the other one would be the HER2 positive.
Yes, yeah, that's right.
Okay, thank you. Thank you very much.
No problem, thank you.
Go ahead, David.
Question about your AIM3.
Yeah.
So these mice are severely immunodeficient. But
Yes.
They have no innate or adaptive immunity. So how do you interpret the data that comes from that in terms of tumor invasiveness?
Yeah, so they have no B, T, or NK cells, but they do have macrophages. And how we interpret it based on the invasiveness is, right now we're trying to look, like the reason we're using immunocompromised mice is so that we can use the human cells and compare it to our human samples. So I mean, with any mouse model where you're using immunocompromised mice, it's very difficult to gauge like the tumor microenvironment. So I guess that is something that maybe we're not assessing fully, but what we're trying to look at is the expression changes in the epithelial tumor cells specifically, so that we can try to compare that to the human. So it's definitely not a perfect comparison, but it's the closest we have right now.
Normal breast epithelium and versus cancer, does that mean?
Yeah, so we're doing it in a couple of ways. Like we're comparing DCIS to IDC, but also normal breast epithelium, like normal benign glands and ducts, and comparing it to DCIS as well. Yeah, kind of like what I showed with my human model, where the DCIS related more to the benign glands than it did to IDC.
You have a question?
Yeah, I just have a quick question
Yeah.
about the DCIS model in terms of the injection there. Do they progress to IDC eventually, or do those cells stay within the?
No, they do progress because we see like full tumors.
Right.
It does take a bit more time because we're injecting like a tenth of the cells that normal orthotopic injections do, but it does progress to whole tumors. And what we're working on is getting the validation that it's IDC versus DCIS. I'm hoping so. So that's why, so I haven't gotten to DESIing the tumors yet, but that's what we're hoping to see. And that's why we're taking mice out every 2 weeks so that we can hopefully target that transition period. Maybe not the direct transition, but like the progression and seeing a change from when the cells are in the ducts to when they gain more of a phenotypic, like a more metastatic phenotype, because the DCIS cells don't have that already.
Oh, that's it. I'm surprised to hear that. So they don't stay DCIS when you engraft them.
Yeah, well, I can't say for sure. It's something that we're working on. I honestly, like, I don't know for sure, but they do kind of, they start to move at, like they take up the whole mammary gland. Like there's not much mammary gland left, so there's not really many ducts left.
So that actually triggers maybe what's the last question here, unless I see another hand pop up. But the samples that you have for your DESI, you say there's 7 DCIS, 3 invasive, and then 10 mixed. So
Yeah.
it made me wonder, at what point do the pathologists decide that it's not mixed, it's exclusively DCIS?
I'm not entirely sure, but I think that it would be exclusively DCIS if you don't see any IDC present in any of the like sections that they're looking at when they diagnose. I'm not entirely sure, but that's what I think they decide. So once they see the presence,
IDC there's no DCIS left there.
There can be IDC with DCIS present, like that's what the mixed is. So like they see samples, like I have some of the samples in here that had DCIS present in the same sample as IDC. So you can see both at the same time, but I think it's classified as mixed once you start to see. I see Dr. Varma has her hand raised, so maybe she has.
Want to weigh in on that, Sonal?
Yeah, so I was just going to comment on that. Most IDCs will have at least some DCIS in there. So it's not that, when you have completely, like when it comes out completely of the ducts, you lose DCIS. And that's something that's very perplexing in terms of how it progresses. But the other thing is that usually with high-grade DCIS, you would see invasion, but it's not that, low-grade DCIS will progress to intermediate to high. It's not like that. There are very different pathways. And usually so it's more common to see a lot of DCIS with low-grade tumors. With high grades, it's weird because the DCIS will be high grade. There will be lots of necrosis and stuff, and then a lot of times it will form a mass, so that's why you're getting it out. It will also have calcs, so both mass and calcs, and that's why it's sort of our data is more enriched for high grade DCIS because it's easier to collect than pure low-grade DCIS. So I guess what I'm trying to say is pure low-grade DCIS, low to intermediate, it's hard to come by. Like you would have maybe one case every 2 months or so of pure DCIS, but high-grade DCIS is easier to come by, and mixed is always, like all IDC will have at least some, not all, but almost all will have some DCIS.
Thanks, Sonal. Sandeep, did you want to weigh in again?
Yeah, thanks, Peter. Yeah, just to pick up where Sonal left off, our center as well was part of a multidisciplinary group with Sunnybrook and others, about 10 or 10 years ago or so, looking at a molecular classification for DCIS. We worked with Eileen Rakovich, who's a rad onc specializing in breast, and the Sunnybrook breast pathologist group there. So there's a lot of work that is going on in this field, and we've been involved with some of that. But from a clinical perspective, I think that the work that you're doing, Natasha, is potentially very important in that, what Dr. Rakovich, the rad onc, was really looking to do is to find at least a subset of patients who could be spared radiation after a diagnosis of DCIS, for example. Because right now, we probably overtreat, we meaning the collective breast caring community, collectively overtreat women with non-invasive breast cancer. And so to find a subgroup, probably through molecular, because I don't think morphology alone does it, that might benefit would be great. And, sort of oncotype is being used somewhat for DCIS too, but we do need other tools to sort of stratify and better predict women at risk or identify those who need treatment more. So I think that's where I think that some of your work could be of more value in the long term. So it's good that you're doing this stuff. Thank you.
Thank you. I really appreciate that.
Thanks, Sandeep. Last chance. Okay, if not, join me in thanking Natasha for an wonderful talk. Thank you.